Now look at this.
What's up about penguins? Today we're going to do topic 4.1 on cell communication. So there's a bunch of different types of cell communication. So we've got autocrine, which is going to be perceiving to the same cell, paracrine, which is going to be local, endocrine, which is going to be long distance, and then direct signaling, where it's going to become in direct contact with another cell. So example of our direct contact, we see this macrophage. The macrophage is going to engulf some type of pathogen. And then on one of its glycoproteins, specifically called the MHC, it's going to present that antigen out. The helper T cell is going to have a receptor that will bind to that antigen. So this is direct signaling because we have a direct contact between these two. So in this picture, we see autocrine signaling at the very top. Auto meaning self. Autocrine means that the ligand or signaling molecule is going to be released out into the extracellular fluid. It's going to bind to a receptor on the same membrane. And so an example of this would be, and we'll talk about a little bit, apoptosis, which is going to be the signal that causes the cell to kind of break down or deteriorate from the inside out. This bottom image has to do with paracrine signaling. Paracrine means that the ligand, signaling molecule, is going to be released out into the extracellular fluid. It's going to bind to a target cell in a nearby area. So how do we know which one's the target cell? Look at the picture at the bottom. These cells down here have more of a kind of rounded receptor, so they bind to the circular ligand. Where the top cells, they have a triangle shaped receptor, so they're not going to bind to that ligand. So you have to have the right receptor to bind to the right ligand because, of course, chemical bonding. In endocrine signaling, this is going to involve that the signal is going to be secreted out and it's going to travel through the bloodstream to a cell somewhere else in the organism's body. So this is like insulin, testosterone, any of the hormones that we talk about. So when we think about that signaling molecule, a lot of the stuff to do with cell communication relies on the polarity of that ligand, okay? So if we look up here at our steroid hormone, that means that the receptor is going to be intracellular. Our ligand is nonpolar and is able to pass directly through that plasma membrane. So we're going to see that the receptor will be intracellular or inside of the cytoplasm or inside of the nucleus. Versus our protein hormone is going to have a receptor on the membrane. It is polar. The protein is polar. So it's not able to pass through that membrane. So the receptor has to be on the membrane in order for it to be able to bind to cause a response. So when we talk about these protein hormones being released, they're going to be synthesized within our rough ER. They're then going to go to the Golgi apparatus for modification, and then they move through a vesicle to bind to the plasma membrane to be secreted. Steroid hormones will be synthesized within the smooth ER. They then go to Golgi for modification, and then they can pass directly through that plasma membrane with no trouble. Remember, if this was helpful, hit that follow button. I'm doing this for every single topic. So if penguin is just success. Bye, y'all.